Our next speaker tonight is Professor Marco Falasca from Curtin University.
Thank you, Paul and Charlene, for your kind invitation. Try to keep my talk very simple, but it's not easy. Do my best to, my passion for what I do for chemistry, for biology and medicine, it will creep in with some chemical formula, so don't get scared. So the main message I want to give you is that, you've already been introduced by Paul and Joe to the field of medicinal cannabis, of cannabidiol, THC, and what I'm talking today is the research we are doing cancer, and in more general, the research ongoing on cancer in the world. So it's what I think is very important, that we need to give some scientific credibility to the use, if any, of cannabis in therapy for cancer, because there are many claims, and I need to give some complexity to my talk, because it's very important to understand the complexity, to appreciate if the claims around are true or false. So, and I want to give you what we do is we have a very interdisciplinary team. I moved from London to Perth in 2014. I've been had a team in London, but I've been able in 3-4 years to establish my own team here in Perth. We have chemists that synthesize a new molecule. We have basic scientists, we have oncologists, surgeons. So it's very important, especially to tackle very challenging cancers. It's very important to have an interdisciplinary group. And, I'm very pragmatic. I'm open to any drug, so that is, if works, is fine. So I'm very open-minded. So what we do, we normally start from patients. We investigate tumors. That is, patients that come from the surgery, and we try to understand what is not functioning, what is more expressed in the patients with cancers, in this case pancreatic cancer, and what we do, we then move to the lab. So we start, it's very important to understand the different levels of complexity, because many of the claims come often from testing drugs of cannabis in very simple models where, you have relatively easy success in killing cancer cells. But the problem is you need to then keep going with the complexity and keeping this activity, anti-cancer activity, with more complex model. So we start from cells, isolated from the tumors of patients in the lab, and then we investigate this specific protein, and then we use models, animal models, mouse. So it's not a good thing for someone, but there is no other way to test and to challenge difficult cancer. So we use animal model, and very complex animal model to in particular, we use models that have a disease that is very similar to the human, because then this is the complexity, because you need to have an effect, a very good effect in these models that have cancers that are very similar to the human, to then have some probability to have success in humans. So, and again, don't get scared by this. We work on the metabolism, but just very briefly want to say that it's now becoming very important to study the metabolism in cancer cells. So cancer cells have a different metabolism. They exploit different routes to produce energy for the cancer cells. So it's very important to tackle this specific altered pathway in the cancer cells that produce energy. And in particular, we are interested in lipids. So when we talk of endocannabinoids, keep in mind that these endocannabinoids, the cannabinoids that we have in our body, are lipids. And these lipids are very similar to the lipids we are studying. So in the last 30 years, I've been interested in studying these particular lipids. That is I've shown that this lipid is accumulated in certain cancer. And so what it does is release in the media and activates cancer cells. So in the 90s, my first studies, I didn't know the receptor. And more recently, I identified this receptor for this lipid that is named GPRT5. Now, this is considered a putative cannabinoid receptor. And this is why I am interested in this field, because I found this receptor present and expressed in high quantity in different cancers. So therefore, my question is, as I said, a very pragmatic approach. I want to stop this. I want a drug that stops the activity of this drug. And long story short, the antagonist, the drug that inhibits this receptor is cannabidiol, among others, cannabidiol. And following my studies, the other group I found that this receptor is overexpressed and have a key role in different cancers. And in normal condition, this receptor is found in the body, in adipose tissues and the highlights, liver, and is involved in energy homeostasis. And you don't have to be a chemist to appreciate the fact that the lipids that activate this receptor are very similar to endocannabinoids, and they can be considered putative endocannabinoids. So I just want to briefly summarize and give so what we call endocannabinoids are molecules that have a cannabinoid structure that are in our body, and they have a function in transmitting signaling and have a role in our physiology in our body. The phytocannabinoids is what is found in the cannabis, for instance, so a very similar chemical structure, but they're found in the cannabis, and when we take these substances, they mimic, they interact with the receptor for the cannabinoids, and this is the way they work. And then we have synthetic, so when in the lab, we synthesize molecules with the same chemistry, so we talk about synthetic cannabinoids. So you've seen already that the main cannabinoid receptor are the CB1, cannabinoid receptor 1, and cannabinoid receptor 2. So the GPRT5 is considered to be another cannabinoid receptor, so it's also involved in these signals, and these are the endocannabinoids. So therefore, the phytocannabinoids intervene on this receptor and interact with this receptor, and their activity is due to this interaction with these receptors. So this is a typical example of how the endocannabinoid system works. So you can have release on endocannabinoids and activity in the specific receptor, and THC or other phytocannabinoid can interfere with this activity by interacting with the same receptor. So you've seen already the main receptor found the CB1 in the brain, on the CB2 in the immune cells around the body. And so, as I said, my main interest is this receptor GPRT5 that is expressed in high quantity in different cancers, specifically in pancreas. So we have a long-standing interest in pancreas. This is a very challenging disease. This is very difficult to treat tumors, and it's very heterogeneous. And if you look at the statistic, at the numbers, so you see that the survival of these patients is very low, and it's highly required to have a new therapy, new strategies to tackle these cancers. And if you look at the survival improvement in compared to other cancers, this is a survival at 5 years, and you see that if you see some tumors we had in the last 40 years, incredible increase in survival and success. Unfortunately, for other cancer, we don't have the same success. And then this is my interest in working this challenging cancer. And so what we found, to make it simple, we found that in the tumor of pancreatic cancer patients, and in our mouse model, there is a higher expression of this receptor, so suggesting that this receptor has a role in the progression of the tumor. And As I said, as a model, we use this mouse that have the mutation of two molecules called KRAS and P53, that are KRAS is 95% of patients of pancreatic cancer have mutation in KRAS, 70% of patients have mutation in P53. So this mimics very well the disease, the human disease. So it's a good model to study therapeutics. So what we found first, we demonstrate that when we eliminate the expression of the receptor, GPFT5, the tumor growth is slowed, is relented in this mouse model. So indicating that GPFT5 is a good therapeutic model. So what we've done next, we tested drugs that blocks GPFT5 in this mouse model, and we use cannabidiol. The reason why we have other potential drug, but they are more toxic, and to get to the clinic is more difficult. So cannabidiol is already in the clinic, may improve the side effects of the chemotherapy. So there are many advantages in use cannabinoids instead of other synthetic chemicals. So this is the survival curve of the animal we treated. So this is normally these mice when they spontaneously develop the tumor, and when the tumor is palpable, normally in like 30 days, all of them die because of the cancer. So we treated them with the normal chemotherapy, and this is the standard a few years ago was gemcitabine, and we don't see much improvement. This is unfortunately what happened in the clinics. So patients do not have, there is a slight improvement in survival and the patient feel better, but unfortunately then the tumor relapse and there's no more activity. So what is important, when we use the cannabidiol alone, we didn't see much activity. We were expecting to see something, we were hoping to see something, but unfortunately there was no improvement. Maybe slight, but nothing significant. But what is remarkable is that when we combine the chemotherapy with cannabidiol, we had a fantastic extension of the survival of these mice. And this is the base of what we now want to move to clinical trials. The clinical trials are under discussion here in Perth, and we hope to have soon result and possibly something very similar, and see the extension of survival of patients with pancreatic cancer with this combination. So the long story short, when we use the cannabinoids, the cannabidiol in this case alone, we don't see improvement, but the combination gives tremendous improvement. So, and this is in the days of survival. In this case, you can see with the combination, we have 3 times increase in the survival of this mice. So, and this is what we conclude from cannabidiol, is there's a weak anti-cancer activity alone, but potentiates a lot of chemotherapy. So what next? We know that cannabis contains many other cannabinoids, terpenes. So our interest is to see if there is this so-called entourage effect. So if many components of cannabis may potentiate themselves, and we can get even a better improvement of chemotherapy compared to cannabidiol alone. And so this is basically what we're doing. And there are other cannabinoids that have been proposed to have anti-cancer activity. So we want to have a scientific validation of this entourage effect and see if we have indeed. Because as a matter of fact, when you don't go to the clinic saying, oh, I found a fantastic therapy. So the clinician want to see hard evidence. And this hard evidence is the complex model that we have. And they say, oh, then this is likely to have something good in the patient. So this is why it's so important to have our preclinical validation of drugs and to have complex model to study these therapeutics. And thanks to Zelda, we are testing now different extracts from enriched either in THC or CBD with different chemotherapeutics, and I hope again soon to have as I said, clinical trials, thanks to Zelda, are now under discussion. We hope to have soon. It's a long process. It's not easy, but it's ongoing. And now the standard of care is gemcitabine plus Abraxane, and this is what we now want to do. Very simple, gemcitabine, because the patients will still be treated, but we will have 2 groups. One is with the chemotherapy plus minus cannabidiol or other cannabis extract, whatever we found more active. So I would just want to conclude that we are in the middle. From one side, our people that claim that cannabis can cure cancer. Absolutely not, there's no evidence at the moment. However, I can say that even on the other side, there is a skepticism, or they say, no, cannabis is not doing nothing at all. Or what we've seen with my study, this is not entirely true, because I think cannabis or cannabidiol or mix of cannabinoids can be valuable tools in therapy, because they can not only improve the chemotherapy, they can also improve the side effects. And I can tell you, just to conclude my talk, that we have seen in our models, in these mice that spontaneously developed, the chemotherapy is extremely toxic. And most of the time, cannabinoids improve a lot the side effects in our models. So I just want to thank my group and Avner, the Pancreatic Cancer Foundation. I want to invite you this Sunday, South Perth, 10 o'clock, there is a walk to support the Pancreatic Cancer Foundation. So please come and join us to support this research. Thank you very much.